Welcome to the topic of the animal endocrine system. It's fitting that we cover this chapter immediately after the nervous system content because the nervous system works closely with the endocrine system to regulate an animal's internal physiological functions. The nervous system sends signals to the cells and glands of the endocrine system. In response, the cells and glands secrete chemical signals called hormones. These hormones are then released into the bloodstream and circulate throughout the body. Endocrine communication is generally slower and more prolonged than the rapid and brief signals transmitted by nerve cells. Whereas some hormones act locally, many others are released into the bloodstream, allowing them to circulate and influence distant target cells throughout the body. Hormones exert specific effects on particular target cells by binding to receptors on or in target cells. The downstream influence of hormones is amplified in a series of steps that occur along a particular endocrine pathway, so that a small amount of hormone can have dramatic effects in the body. In conjunction with the nervous system, the endocrine system regulates an organism's response to the environment. Changes in the environment often present physiological challenges that an organism must respond to by altering the functional state of its body. Such environmental changes could be changes in light or temperature, the threat of a predator, or the presence of a potential mate. Any of these changes stimulates an animal's nervous system and endocrine system to respond. This lecture will give us a closer look at how this works. Endocrine control of internal body functions is central to homeostasis. Organisms have physiological mechanisms that maintain a stable internal environment. Without them, changing environmental conditions would lead to dangerous shifts in an animal's physiological function. For example, an animal's body weight depends on the regulation of its energy intake relative to energy expenditure. Hormones help regulate appetite and food intake. Disruption of these hormones can lead to food disorders that promote obesity or lead to weakness and lethargy. In another example, hormones regulate the concentration of key ions in the body, such as sodium and potassium. Proper ion concentrations are fundamental to healthy nerve and muscle function and to fluid balance within the body. The body is also able to maintain homeostasis by releasing hormones. Hormones are released in response to stimuli and have an effect on distant organs. Maintaining homeostasis depends on feedback from the target organ sent back to the endocrine gland. In this feedback, a stimulus evokes a response, and that response in turn affects the stimulus. Thus, feedback is the means by which a signal produced by the regulatory organ can modify its own subsequent production. Homeostasis typically depends on negative feedback. In negative feedback, a change in a system causes a response that brings the system back to the starting point. The maintenance of homeostasis is an active process. For example, the amount of glucose in the blood of animals is maintained at a steady level. If glucose levels are too low, cells of the body do not have a ready source of energy. If glucose levels are too high for too long, they can damage organs. Maintaining steady blood glucose levels is challenging because glucose is absorbed into the bloodstream from the intestine after a meal, and glucose is readily taken up by cells to meet their energy needs. How then are constant levels maintained in the body? First, after a meal, blood glucose rises. In response to this stimulus, beta cells of the pancreas secrete the hormone insulin, which circulates in the blood. In response to insulin, muscle and liver cells take up glucose from the blood and convert it to a storage form called glycogen. This process results in a decrease in blood sugar, which in turn stops the pancreas from secreting more insulin. So what happens if blood glucose levels fall too low several hours after a meal? First, a different population of cells in the pancreas, called alpha cells, secrete the hormone glucagon. The effects of glucagon are opposite to those of insulin. Glucagon stimulates the breakdown of glycogen into glucose, and it's released from muscle and liver cells. The result is that blood glucose levels rise. This rise in blood sugar causes the pancreas to stop secreting the hormone glucagon. In both cases, the stimulus, either high or low blood glucose levels, is sensed by cells of the pancreas, the beta or alpha cells, and that triggers a response, the secretion of insulin or of glucagon, bringing blood glucose levels back to the set point. When the control of blood glucose levels by insulin fails, a disease called diabetes mellitus results. When untreated, diabetic individuals excrete excess glucose in their urine because blood glucose levels are too high. Diabetes also causes cardiovascular and neurological problems, including loss of sensation in the extremities, particularly the feet. In some instances, it is necessary to enhance the production of a hormone, accelerating the response of the target cells for a period of time. Positive feedback provides this enhancement. In positive feedback, a stimulus causes a response in the same direction as the initial stimulus, which leads to a further response, and so on. We saw this in the nervous system with the creation of an action potential. In positive feedback in the endocrine system, a stimulus leads to secretion of a hormone that causes a response, and the response causes the release of more hormone. The result is an escalation of the response. A positive feedback loop continues to reinforce itself. It can be interrupted or broken only by some sort of external signal. An example of positive feedback occurs in mammals during childbirth. In response to uterine contractions, the stimulus, the hormone oxytocin is released from the pituitary gland, which serves as the sensor. The oxytocin, the effector, stimulates the uterine muscles to contract more forcefully and more frequently. This is the response. Uterine contractions, in turn, stimulate the pituitary gland, the sensor, to secrete even more oxytocin, the effector, causing the uterine muscles to contract still more forcefully and more frequently, the response, with the end result being the birth of the child. Hormones play diverse roles in the body, enabling regulation and coordination of multiple bodily functions in response to environmental cues. Because hormones are released into the bloodstream, they have the potential to affect many organ systems. Hormones are grouped into three general classes shown here: hydrophilic peptide hormones, hydrophilic amine hormones, and hydrophobic steroid hormones. Peptide hormones and amine hormones are both derived from amino acids. Peptide hormones are short chains of amino acids, whereas amine hormones are derived from a single aromatic amino acid, such as tyrosine. Peptide hormones are sometimes large enough to be considered proteins. All steroid hormones are derived from cholesterol. Another important difference is that where peptide hormones can evolve through changes in their amino acid sequence, steroid hormones cannot. Receptors on target organs bind blood-borne hormones, allowing the hormones to influence the function of specific cell targets. Peptide and amine hormones are more abundant than steroid hormones and are more diverse in their actions. Because most peptide and amine hormones are hydrophilic, they cannot diffuse across the plasma membrane. Instead, nearly all peptide and amine hormones bind to membrane receptors on the surface of the cell, triggering the intracellular secondary messenger pathways. Peptide and amine hormones activate or inactivate enzymes within the cell. Typically, they affect protein kinases, which energize other proteins through phosphorylation. This initiates signaling cascades within the target cell. These cascades can lead to changes in gene expression or metabolism, trigger a cell to grow, divide, or change shape, or lead to the release of other hormones. Peptide and amine hormones act on timescales of minutes to hours. In contrast, steroid hormones are hydrophobic. They diffuse freely across the cell membrane to bind with receptors in the cytoplasm or nucleus, forming a steroid hormone receptor complex. Hormone receptor complexes that form in the cytoplasm are then transported into the nucleus of the cell. These complexes most commonly act as transcription factors. That is, they stimulate or repress gene expression, and therefore alter the proteins produced by the target cell. Consequently, steroid hormones typically exert profound and long-lasting effects on the cells and tissues they target, with timescales of days to months. The actions of steroid hormones, therefore, tend to have a broader impact on body function than those exerted by amine and peptide hormones. Your text includes diverse examples of major vertebrate hormones. While I certainly encourage you to explore the full list, you will only be accountable on exams for the details that we cover in this lecture. Let's begin with peptide hormones. In vertebrates, growth hormone, released by the anterior pituitary gland, stimulates protein synthesis and growth of many body tissues, particularly the musculoskeletal system. Insulin and glucagon are peptide hormones released by the pancreas. They regulate glucose metabolism by the liver, muscles, and other tissues. Gastrin and cholecystokinin regulate mammalian digestive function. Gastrin is released by the same organ that it impacts, whereas cholecystokinin travels from the small intestine to other organs that play vital roles in digestion. In addition to serving as neurotransmitters released by the sympathetic nervous system, epinephrine and norepinephrine, also known as adrenaline and noradrenaline, act as amine hormones, supporting an animal's fight or flight response to stress. These hormones are released from the adrenal glands found above the kidneys. Shifting gears to the steroid hormones, the sex hormones estrogen, progesterone, and testosterone regulate the differentiation, maturation, and functional state of the vertebrate reproductive organs. Cortisol, another hormone released by the adrenal gland, mediates a vertebrate animal's response to stress and inhibits inflammation. Steroid hormones similar to cortisol are used medically to reduce the symptoms of certain inflammatory disorders. However, they carry risks because they also depress the immune system, increasing the chance of infection and diminishing wound healing. Hormones are typically released in small amounts, but then they're usually amplified. Amplification occurs during a series of signaling steps in a hormonal pathway, initially between different endocrine glands and then within the target cells after the hormone binds to a cell receptor. Signals are thus amplified at each step of the pathway, resulting in a large effect on the target organ system. Hormonal signaling pathways between endocrine glands and tissues are often referred to as endocrine axes. For example, in vertebrates, the hypothalamic pituitary axis is a signaling pathway from the hypothalamus to the anterior pituitary gland, and then from the anterior pituitary gland to target glands of tissues in the body. Hormone signals are amplified at multiple steps in this pathway. First, trace amounts of peptide hormones, called releasing factors, are initially released by the hypothalamus. Then, those releasing factors signal the anterior pituitary gland. This leads to a much larger release of associated hormones from that organ. For example, corticotropin releasing factor secreted by the hypothalamus stimulates the release of a larger amount of adrenocorticotropic hormone, ACTH, by the anterior pituitary gland. Hormones released by the anterior pituitary gland then bind cell receptors in the target organ. In this case, ACTH acts on cells of the adrenal cortex, stimulating them to secrete the hormone cortisol. Cortisol acts on many different cells and tissues in the body, causing what is known as an acute stress response. Among its effects, it causes the liver to convert glucose to glycogen. This action yields 56,000 times more glycogen molecules than the initial number of releasing factors secreted by the hypothalamus. Here we see an overview of the endocrine organs and their hormones and where they are in the human body. Most hormones have an ancient evolutionary history, and the structure of many hormones are evolutionarily conserved. Typically, the same hormone serves different functions in vertebrates and in invertebrates, and may even serve different functions within distinct groups of vertebrates. For example, the thyroid stimulating hormone, TSH, by the anterior pituitary gland and targets the thyroid gland. In vertebrate animals, it regulates metabolism. Additionally, it triggers metamorphosis in amphibians and feather molt in birds. It has even been found in snails and other invertebrates that lack a thyroid gland. Recent genomic analysis has revealed that the receptors for many hormones evolved well before the hormones with which they now interact. Even though the structure of a hormone or its receptor is often largely unchanged across diverse groups of organisms, the functions of hormones and their receptors can readily be selected to take on new roles. Hormones achieve specificity by binding to receptors on the surface of or inside the target cells. Therefore, it is the presence or absence of a receptor for a given hormone that determines which cells respond to the hormone and which ones do not. The binding of a hormone to its receptor leads to changes in the target cell, resulting in a cellular response. The specific action of a hormone depends on the kind of response that it triggers in the target cell. Another intriguing finding is that many peptides originally identified as hormones in the various tissues also have been found to function as neurotransmitters in the nervous system. Gene expression of a hormone in one kind of cell may produce the same compound but for an entirely different function from gene expression of that same hormone in another cell. The role of hormones as chemical messengers are varied within an organism and easily change over the course of evolution. I've split the image of the human endocrine system so that we may better investigate the details. The vertebrate endocrine system regulates changes in the animal's physiological and behavioral states in response to sensory cues. The nervous system receives sensory cues both from the environment and from internal organs. Sensory signals are processed within the brain and transmitted to the endocrine system primarily by the hypothalamus, which is located in the forebrain. The hypothalamus relays signals to the pituitary gland, which lies just below it. The pituitary gland is a central regulating gland, releasing hormones that coordinate the action of many other endocrine glands and tissues. These glands control the growth and maturation of the body, regulate reproductive developmental functions of the animal, coordinate digestion and metabolism, and control water balance. These effects reflect the broad range of roles that the endocrine and nervous systems perform in maintaining homeostasis. The pituitary gland communicates with many cells, tissues, and organs of the body. Some pituitary targets, like the thyroid and adrenal glands, only secrete hormones and therefore have exclusively endocrine functions. Others, like the lungs, kidneys, and digestive tract, harbor endocrine cells that secrete hormones and also have other physiological roles in the body. Consequently, the vertebrate endocrine system is not localized in one part of the body, but is present throughout, as we can see here. The hypothalamus is the main route by which nervous system signals are transmitted to the vertebrate endocrine system. The job of the hypothalamus is to transmit these signals to the pituitary gland, which is the endocrine gland that acts as a control center for most other endocrine glands in the body. The pituitary gland is divided into anterior and posterior regions. These two regions have distinct functions and organizations. They also have distinct embryonic origins. The anterior and posterior pituitary glands receive input from the hypothalamus in different ways. The hypothalamus contains a type of neuron called a neurosecretory cell. Instead of secreting neurotransmitters that bind to another neuron or to muscle, neurosecretory cells release hormones into the bloodstream. Some of these neurosecretory cells communicate with the anterior pituitary gland. In this case, they secrete hormones called releasing factors into small blood vessels that travel to the anterior pituitary gland. The posterior pituitary gland contains the axons of neurosecretory cells whose cell bodies are located in the hypothalamus. These axons release hormones directly into the bloodstream. Consequently, the posterior pituitary is part of the nervous system itself. In response to signals from the hypothalamus, distinct hormones are secreted by the anterior and posterior pituitary glands. The anterior pituitary gland hormones include TSH, or thyroid stimulating hormone, which acts on the thyroid gland. Also includes the gonadotropic hormones, follicle-stimulating hormone FSH and luteinizing hormone LH, which act on the female and male gonads, the ovaries and testes, and the adrenocorticotropic hormone ACTH, which acts on the adrenal glands. In each of these cases, the anterior pituitary hormone acts on an endocrine gland to cause release of other hormones. Hormones that control the release of other hormones are called tropic hormones. For example, in response to TSH, the thyroid gland releases thyroid hormones that regulate the metabolic state of the body. In response to FSH and LH, the ovaries release estrogen and progesterone, and the testes release testosterone. In response to ACTH, the adrenal glands release cortisol, which has diverse effects that include stimulating glucose release into the bloodstream, maintaining blood pressure, and suppressing the immune system. The anterior pituitary gland also secretes growth hormone, or GH, which stimulates the growth of muscles, bones, and other body tissues, and prolactin, which stimulates milk production in the breast of female mammals in response to an infant's suckling. The posterior pituitary gland hormones include oxytocin and antidiuretic hormone, or ADH, also called vasopressin. These are two evolutionarily related peptide hormones, each consisting of 9 amino acids. Oxytocin plays several roles related to female reproduction. It causes uterine contraction during labor and stimulates the release of milk during breastfeeding. Vasopressin, or ADH, acts on the kidneys, regulating the concentration of urine that an animal excretes. This is critical to maintaining water and solute balance in the body. Some of the hormones released by the anterior pituitary gland act on endocrine organs that then release hormones themselves. These tropic hormones are TSH, FSH and LH, and ACTH. TSH acts on the thyroid gland, which is located in the front of the neck. The thyroid gland releases 2 peptide hormones, thyroxine, or T4, and triiodothyronine, T3. These hormones regulate cellular metabolism throughout the body. Overproduction of thyroid hormones, or hypothyroidism, creates symptoms that reflect an overly active metabolic state, such as increased appetite and weight loss, whereas thyroid hormone deficiency, or hypothyroidism, creates symptoms that reflect a metabolic state that is too low, such as fatigue and sluggishness. Both conditions are diagnosed by blood tests that monitor circulating levels of thyroid hormones, and both can be treated by medication. Thyroid hormones require iodine for their formation. Individuals who do not acquire enough iodine from their diets produce too little thyroid hormone. Additionally, iodine insufficiency stimulates increased production of TSH by the anterior pituitary gland because of the absence of negative feedback. Over time, the thyroid gland enlarges to form an enlargement of the throat called a goiter to compensate by producing more thyroid hormone. The introduction of iodized salt has eliminated goiter formation and hypothyroidism in many countries, but in many underdeveloped areas, these remain significant public health problems. The gonadotropic hormones are FSH and LH. They target the female and male gonads, the ovary and testis. In response to FSH and LH, the ovary and testis each secrete sex hormones that regulate their own development, as well as the sexual differentiation and maturation of secondary sexual characteristics of the body. Female sex hormones include the steroids estrogen and progesterone. The principal male sex hormone is the androgen testosterone. Testosterone is a naturally occurring anabolic steroid. Anabolic steroids promote protein synthesis to build body tissues and anabolic metabolism to store energy within cells. Testosterone stimulates the synthesis in the testes of proteins needed for sperm production. Elsewhere in the body, it stimulates the development of male sexual features and body tissue growth, particularly in muscles. A variety of synthetic anabolic steroids have been developed to treat muscle wasting due to loss of appetite and diseases such as cancer and AIDS. Anabolic steroids are used illegally by both male and female athletes to promote muscle strengthening. The risks to health are considerable. They include liver damage, heart disease, and high blood pressure. Tissue damage occurs because elevated circulating levels of testosterone inhibit the normal synthesis of anterior pituitary tropic hormones. ACTH is also a tropic hormone released by the anterior pituitary gland. This hormone acts on the cortex, or the outer portion, of the paired adrenal glands. In humans, each small adrenal gland lies just above each kidney. ACTH stimulates adrenal cortex cells to secrete cortisol during times of stress, such as starvation, fear, or intense physical exertion. Although many endocrine organs receive signals from the hypothalamic pituitary axis, some respond instead to internal physiological states of the body. One of these, adjacent to the thyroid gland, is the parathyroid gland. This gland secretes parathyroid hormone, or PTH. Calcitonin, which is secreted by the thyroid gland, and PTH together regulate the activity of bone cells that control bone formation and bone removal, and therefore the levels of calcium in the blood. When circulating levels of calcium fall too low, the parathyroid gland triggers release of PTH. That hormone stimulates osteoclasts to reabsorb bone mineral, halting bone formation and raising blood calcium levels. When calcium levels are too high, the production of PTH is inhibited. Calcitonin instead is released to shift bone metabolism toward net bone formation, building bone that stores calcium in the skeleton. Note that, as with blood glucose levels, calcium levels are maintained in a narrow range by negative feedback. The effect of the hormone, increasing or decreasing levels of calcium in the blood, is the opposite of the stimulus, low or high levels of calcium in the blood, so that a stable set point is maintained. Whereas hormones enter the bloodstream to be transmitted to more distant target cells, as shown here on the left, other chemical compounds act locally on neighboring cells, as shown to the right. These cells have receptors that bind to the signal compound before it degrades or diffuses into the bloodstream. Chemical compounds that act locally are said to have paracrine function. If the compounds act on the secreting cell itself, they are described as having autocrine function. That is, the chemical signals stimulate or inhibit their own secretion. Growth factors and histamine are examples of paracrine chemical agents released in small amounts that act locally on neighboring cells. Locally acting signals have immediate short-term effects because they are quickly degraded. Many animals release small chemical compounds into the environment called pheromones to signal and influence the behavior of other members of their species. Sex pheromones play a key role in the mating behavior of many invertebrate and vertebrate species. In most mammals, amphibians, and reptiles, pheromones are detected by a femoral nasal organ with chemosensory neurons in the nasal region of the skull. The release of sex pheromones is one of the most common types of pheromone signaling used to attract a mate and trigger ritualistic social and mating behaviors. Other pheromones signal territorial boundaries or predatory threats. Dogs and other canids, as well as cats, secrete pheromones in their urine that mark their territories. Ants and other social insects commonly deploy alarm pheromones to warn of an attack from another colony. The release of alarm pheromones from a stinging bee causes other bees to swarm and join the attack to defend their nest. Social animals such as deer warn of an approaching predator by means of alarm pheromones. This brings to a close our discussion of the animal endocrine systems.